Hello students, today we can see the topic cell wall. As we have seen earlier, the cells of all the organisms are surrounded by a plasma membrane, isn't it? But in some of the cells, that is the plant cells or fungal cells or even the bacterial cells, they are surrounded by an outer covering called the cell wall. Okay, in addition to plasma membrane, the plant cells are surrounded by another rigid outer covering called the cell wall. Okay, the cell wall can be seen outside the plasma membrane. Okay, the cell wall is present outside the plasma membrane. This cell wall is made up of cellulose. So what the cell wall is formed of? It is composed of cellulose. Okay. Now what is the cellulose? It is a complex substance, a complex sugar that provide structural strength to the plants. Okay. So what is cellulose? Cellulose is a complex substance, or we can say it is a complex sugar that provide structural strength to the plants. Okay, now we can see what is plasmolysis. Earlier we have seen that when a plant cell is kept in hypotonic solution, it will absorb water. That is, water enters into the cell and the cell will swell, isn't it? On the other hand, when we keep a plant cell in hypertonic solution, the water will be lost from the cell, isn't it? The water will be moving from the cell to the solution. Why? You know that always the water will be moving from a region of its higher concentration to a region of its lower concentration, isn't it? So, when a living plant cell loses water through osmosis, there is a shrinkage of the contents of the cell away from the cell wall. This phenomenon is known as plasmolysis. So what is plasmolysis? When a living plant cell loses water through osmosis, that is, if the surrounding medium of the cell of that plant cell is having hypertonic solution, then what will happen? The plant cell will be losing water. How it loses water? By the process of osmosis. So from where to where the water will be moving? The water will be moving from the cell to the solution. Why? Because the cell will be having more amount of water and the outer medium or the solution will be having less concentration of water compared to the cell. So automatically the water will be moving from the cell to the surrounding medium. Okay. So when a living plant cell loses water through osmosis, there is a shrinkage or contraction of the contents of the cell. So whatever is present inside the cell, all that will be contracting and they will be moving away from the cell wall. This phenomenon is known as plasmolysis. Okay. So if you see this picture, you can see here the outer cell wall is present on the outer side, and the green colored structure which you are seeing is a layer of protoplasm. In the center, nothing is there. Actually, that is the large vacuole. Okay. Now, when this cell is kept in this hypertonic solution, you can see slowly the contents of the cell is moving away from the cell wall. Isn't it? Here also you can see that cell wall can be seen clearly. But if you see in the first picture, you cannot differentiate between the cell wall and the protoplasm. Both are attached together, isn't it? But when they continuously lose the water, what happens? All the water will be lost from the cell. As a result, the inner cellular contents have moved away from the cell wall. They have moved away from the cell wall, and this phenomenon is known as plasmolysis. Okay. How the plant cells can withstand very dilute external media without bursting, or how the plant cells can withstand greater changes in the surrounding medium than animal cells. The answer for that is the cell walls permit the cells of plant or fungi or bacteria to withstand very dilute external media without bursting. How it occurs? When the plant cell is kept, plant cell or it is a bacterial cell or a fungal cell, when it is kept in a hypotonic solution. You know what is hypotonic solution? The solution which is having more amount of water than the cell. So when the plant cell is kept in a hypotonic solution, what happens? The water will be entering into the cell from the solution, isn't it? By the process of osmosis. Okay. So the water enters into the cell by osmosis. When more and more water enters inside the cell, what happens to the cell? The cell will swell up. Okay. The cell swells and it creates a pressure against the cell wall. The swollen cell will be creating a pressure against the cell wall because now more water has entered. As a result, it will create a pressure and that pressure will be created against the cell wall. Okay. Now, the cell wall in turn exerts an equal pressure against the swollen cell. Okay, the cell wall is rigid in nature, isn't it? It is not very thin. It is hard structure. It is rigid in nature. So this cell wall will exert an equal pressure against the swollen cell. Okay. You have studied that every action has equal and opposite reaction, isn't it? So when the cell is creating a pressure against the cell wall in turn creates a pressure against the swollen cell. Okay. How it is done? It is due to the presence of the thick cell wall. If there is no cell wall, what will happen? When a pressure is created against the outer covering, that covering may burst, isn't it? But here, due to the presence of the rigid cell wall, it can create an equal pressure against the swollen cell. Okay? Because of the presence of the cell wall only, the cells can withstand much greater changes in the surrounding medium than the animal cell. So the plant cells or fungal cells or bacterial cells, because of the presence of their cell walls, they can withstand much greater changes that occurs in the surrounding medium than the animal cells. Okay. When we take an onion peel and when we see it under the microscope, we are able to see the cell, isn't it? Can we see it clearly? No, it will be transparent. Okay, it will be colorless. So, before seeing any cell under the microscope, we have to stain it. What is that staining? It is adding a coloring material to the cell so that the different part of the cell will absorb that coloring material, that is the stain, and it stains or it gets colored differently. Okay, some region of the cell gets darkly colored and some regions will be lightly colored. Okay, so what such stains can be used? For plant cell, we can use iodine solution or even safranin, and normally for animal cells, we can use methylene blue. It is not that only methylene blue can be used for staining animal cell and safranin can be used for staining plant cell. It is not like that. Okay, for different cells, different type of stains can also be used. If you take iodine solution, that can be used for staining onion peel. At the same time, onion peel can also be stained by safranin. Okay, so when we add the stain, what happens? Different part of the cell will be staining in differently. That means some part will be darkly stained and some will be lightly stained or lightly colored. So the darkly colored structure which you can see in the cell towards the center of the cell is the nucleus. Okay, so when you stain a cell and see it under the microscope, the darkly colored structure which you can see towards the center of the cell will be the nucleus. Okay, so the nucleus may be spherical or oval or dot-like structure inside the cell when you see it under the microscope. Okay. Now, generally, each cell will have one nucleus. Okay, only one nucleus will be there in each cell. But there are certain exceptions. In some of the cells, more than one nuclei will be seen. Okay, nuclei is plural, nucleus is singular. Okay, for example, if you take this striated muscle, it will be having more than one nuclei. Okay, and some of the fungal hyphae, if you see also, there also we can see more than one nuclei will be present. Okay, but generally each cell will be having one nucleus in it. Okay, now who discovered this nucleus? Nucleus was discovered by Robert Brown. Already you have studied that nucleus was discovered by Robert Brown. Okay, in the year 1831. When we see the structure of the nucleus is surrounded by a double layered covering called the nuclear membrane or the nuclear envelope. Okay, so here you can see it is a double layered membrane. Okay, this double layered membrane is called the nuclear membrane, or we can say it as the nuclear envelope. And this nuclear membrane is having pores. Pores means what? Holes, which allow the transfer of material from inside the nucleus to the outside. Okay, the nuclear membrane is not continuous. It is having pores. You can see here, this is the nuclear pore. Okay, and what is the use of this nuclear pore? It allows the transfer of materials from inside the nucleus to its outside, and sometimes from outside to the inside also. Okay, now within the nuclear membrane, that is inner to the nuclear membrane, a matrix will be present, a fluidy material will be present inside the nucleus, and that is called the nucleoplasm. The fluid which is present inside the cell, we are saying it as cytoplasm. Same way, the fluid which is present inside the nucleus is called nucleoplasm. Okay. Nucleus contains chromosomes. So where the chromosomes are present, they are present inside the nucleus. So what is this chromosome? These are rod-like structures which can be seen inside the cell when the cell is about to divide. Okay, so when a cell is going to divide, we can see some rod-shaped structures in the nucleoplasm or inside the nucleus, and these are said to be as the chromosomes. Okay, it contains, that is, this chromosome contains information for inheritance of characters from parents to the next generation or to the offsprings. Okay, so the chromosome contain information for inheritance. Inheritance means transferring from parent to the offspring. Okay, offspring means the next generation, the young ones. Okay, so chromosome contains information for inheritance of characters from parent to the offspring in the form of DNA. So what is present in the chromosome? DNA is there. So chromosomes are composed of DNA and protein. What is DNA? If DNA is a deoxyribonucleic acid, and that DNA is responsible for transmitting the characters from the parent to the offspring. Okay, the DNA molecule contains the information which is necessary for constructing or forming a new cell and organizing the cell to control the cell. So the DNA molecule is having all the information which is necessary for constructing a cell, for making a new cell and organizing the cell. Okay, controlling the activities of the cell. Okay, the DNA is made up of the functional unit called the genes. Okay, the functional segment of DNA are called genes. Actually, It is a small segment of the DNA. DNA is a very long structure, and it is divided into many small segments, and each segment is said to be as a gene. Okay, don't think it is divided into pieces. It is not like that. In that long DNA, certain segments are considered as genes. So genes are responsible for controlling our characters. Each and every organism is having their own specific characters, isn't it? These characters are controlled by the genes, just like the eye color, the skin color, the hair, whether it is long or curly, like that each and every character is controlled by the gene. Okay. Now, what is the difference between chromatin and chromosome? What is chromatin and what is chromosome? I told you chromosome is a rod-like structure which is seen in the nucleoplasm or inside the nucleus when a cell is about to divide, isn't it? Now, what is chromatin? The same chromosome is said to be as chromatin in a non-dividing cell. That means when a cell is not dividing, the DNA is present as a part of the chromatin material. Okay, so what is that chromatin? It is an entangled mass of thread-like structure. What do you mean by that entangled mass of thread-like structure? It is a highly coiled thread-like structure. Okay, so the chromatin material will be seen as very long, thin, thread-like structure which are coiled with each other. Okay, so that is said to be as chromatin. Okay, so in a non-dividing cell, the DNA is present as a part of the chromatin material, and this chromatin material can be seen as entangled mass of very thin thread-like structures. Okay, but when the cell is about to divide, this chromatin material gets organized into chromosome. That is, this chromatin material gets highly coiled, closely coiled. As a result, it becomes a rod-like structure. In the picture, you are able to see this rod-like structure, isn't it? Now it becomes a rod-like structure, and this structure is said to be as the chromosome. Actually, chromatin and chromosome are the structures which are responsible for inheriting the characters from parent to the offspring. Okay, so both are same only. Slight difference is there in their structure. Okay, in a non-dividing cell, the structure which is present is the chromatin material, which will be very long, thin, thread-like structure which is seen as entangled mass. Okay, but when you see a chromosome, it will be seen only in the dividing cell, where this chromatin material will become closely coiled, very highly coiled, which are very close to each other. As a result, it becomes a rod-shaped structure. Is it clear? Next is the function of nucleus. The nucleus performs mainly two functions. What are the two main functions performed by the nucleus? One is it plays a central role in cellular reproduction. What do you mean by that cellular reproduction? It is nothing but cell division. Okay, so it controls the cell division. It decides how the cell has to divide, when it has to divide, and to form two daughter cells. Okay, so nucleus plays a central role in cellular reproduction. Now the second point is it directs the chemical activities of the cell. That means it determines how the cell has to develop and in which form they have to develop and how it should exhibit at maturity. That is, what type of cell it should become at maturity. So all these can be directed by the nucleus. Okay, so the nucleus will be directing all the chemical activities that are taking place inside the cell, so that it determines how the cell should develop and what it should become, which type of cell it should become. That is decided by the nucleus. Okay.